undergraduate students at Wheaton College. I'm just here to tell you a little bit about how we're addressing some challenges we see in science education, and to give you an idea of, how you might adapt some of these strategies yourself. And I want to thank you so much for your patience, of course, with the language barrier. And so if you don't mind, I'll just start showing you a few slides. Let's see here. I can find my slides. Here it is. Front. There we go. And I hope that's visible to everyone. Please say so if it's not visible. Okay. Before we start, I'll introduce myself. I have been working at Wheaton College for 3 years now. Wheaton College is in northeastern United States. It's a beautiful college. We have, it's a place where there are there's a lot of snow in the winter. So it can be really very beautiful in the snow. And I teach many biology classes. So at Wheaton, I've taught biology introduction. I've taught microbiology several times. I teach a class called Research Experience, which I'm going to talk about. I teach biostatistics, seminar for seniors, and microbial evolution. So I've been, have quite extensive experience teaching undergraduates biology and biology-related courses. And my department and our college is facing some challenges with science education. And these are really widespread challenges. The big challenge that we're facing right now is that some students are better represented in science classes and in science careers than others. And the bias is against people who don't have a lot of power in society. So ethnic and racial minorities, students whose family members did not go to college, did not study science, students who have economic challenges, who maybe have trouble affording their education or the expenses associated, gender minorities, although I should note that at least in the United States, women are well represented in biology. Other gender minorities and sexual minorities are not, but women are. And that these challenges are kind of related to these processes of recruitment, which means who decides to go into science to begin with, retention, who can stay in science or who switches to another field or perhaps leaves college altogether. And these are related to a sense of belongingness, this feeling that I belong in science, that science is a place for me. And so our college applied for a grant to work on these particular challenges. And then we got this grant. And so all of the work I'm going to tell you is curriculum that was developed by myself or by my colleagues using support from this grant. And the goals of this grant were to improve student outcomes in general, and specifically to provide new opportunities in science, technology, and engineering and mathematics learning. And we abbreviate that STEM. That we give students opportunities to be science leaders, and that we promote inclusivity and inclusive excellence, so that we make science a place where every student feels as though they belong, in hopes of improving recruitment and retention in STEM. And we've decided, especially in the biology department, to use experiential learning to address many of these challenges. So experiential learning is learning in which the student experiences being a scientist in the classroom. And there are studies on experiential learning, and these studies have shown that experiential learning tends to increase the sense of belongingness. I belong in science. I am the kind of person who can be a scientist or who can work in science. Experiential learning tends to build community. And importantly, students are given the opportunity to contribute their own experience, whatever that diverse experience might be. Experiential learning tends to rely less on prior classroom knowledge. So many instructors have this experience where a student who's taken another related class does really well in the class that you're teaching. And we see that a lot in college. So students whose high schools had a lot of resources, the high school has given them a really good biology education. They do very well in introductory biology because they kind of already know things. Experiential learning will even that out a bit because it's new for everyone. And this encourages problem solving, students take responsibility, and science becomes a practice. It's something that you do. It's not a list of facts that you remember. So this is really what we're kind of aiming for. And I'm this whole talk, I put in some pictures of students in our experiential learning classrooms. Everything I'm going to talk about is happening in a lab classroom. And so our goals with these changes and with developing these courses are to increase the students' personal identification as scientists. We wanted to increase their confidence, their access to science, support and opportunities, especially for students who don't normally get opportunities. And an important goal, but a goal that comes after these first four, is to develop these topic-specific technical and conceptual skills. So we want them to do this, but we're really pushing very hard for these other goals of identification and confidence and access to science. And I'm going to give you 3 examples of these. I tried to choose examples from student courses where students were just coming out of high school, because I understand a lot of people in this course are planning on working with high school students in the future. So I'm going to talk about our college introduction biology labs, a course that we call research experience and science communication, and microbiology lab. And the target student populations are all first and second year students. So we'll start out with introductory biology lab. This lab was paired with a lecture course on the introduction to biology. And it had 2 projects. The entire semester focused on these 2 projects. The first project is ecological, and the second one is physiological. And we timed them in such a way where the ecological one happened before the first snow, because students are going to be outside. And you can see in the photograph, not only are they outside, but they're in water. And so in this first question, they're asking about how the ecology of diversity in a pond changes with different chemicals. So they go out to the pond, they measure the chemistry, and they count the diversity. In the second project, they ask how you can change an animal's physiology in the lab. Well, let me give you a few details about this. In both of these projects, the really important thing in the student process is that students form and test hypotheses. So what they're doing is they're practicing the scientific method, but they have to apply their knowledge of biology when they form their hypothesis, and they have to justify it based on the information they're learning in the classroom and based on information that they seek out from outside of the classroom. So this is perhaps the most important thing we have students do, but they're also going to design experiments that test their hypotheses. And because it's introductory, in the first project of the semester, the professor is much more involved than for the second project. For the first project, the professor decides what data will be collected, but students then choose among those data which data can help them test their hypothesis, and they plot those data. With the second project, the students design the entire experiment, and they have to have their hypothesis in mind. And again, they're thinking about the concepts they're learning in the class and how to apply those concepts to their project. So we don't, in introductory biology, have labs where we say, today we're going to demonstrate this. I'd like you to set up this with this protocol and record your results. Instead, we say, I want you to ask how the diversity in the pond changes. You're going to collect some data, but you're going to decide which data are important for testing your hypothesis. So what these students do is they go out to the pond and they measure different different aspects of the chemistry: nitrogen, phosphorus, oxygen, pH. They then take samples and they count different kinds of living things. And we ask them how do they expect biodiversity to correlate with chemistry? And the important thinking thing here for the students is to justify why. If you were to do this with your own students, there's a lot of flexibility to this sort of thing. So you can set it up with a very specific goal. If you have students who are very young, you might want to say, I'm going to give you the hypothesis, but I want you to think about why that might be. You can have them test something very simple like pH, which can require, some pH strips or some kind of indicator, or you can get very fancy and measure, I don't know, the amount of carbon dioxide that's produced or something. In our case, we used a lot of chemical kits. So we would buy a kit that would measure the amount of ammonia dissolved in the water, the amount of phosphorus dissolved. And then we would have the students collect living things that you can see. So invertebrates from underneath, that in fact is what these students are collecting here with the big nets, is the invertebrates at the bottom of the pond. Also, we could have them look at things under the microscope. So it depends on what resources you have. So the students ask their own question and they answer it. And here's an example of a student group. This is a plot that our students produced, where they plotted the relationship between oxygen concentration and the diversity of pond microorganisms. And they found that, in the data that we collected, there was an increase in the number of pond microorganisms as the amount of oxygen increased in the pond. And together as a class, we talked about why they observed that. Now, one thing I should tell you is that, excuse me,
can I ask you something?
Yes.
How do you measure oxygen levels? Is that something that you can easily do, or do you need specialized equipment?
How did we do that?
Estoy preguntando si necesita equipamiento especial para medir oxígeno, como digamos algo caro, o es algo posible de obtener fácilmente.
So if I remember right, we had a kit that would measure dissolved oxygen. So we bought a commercial kit. And what we were specifically looking at was the amount of oxygen that was available for consumption. So we would measure oxygen and then wait a week and measure it at the end and see how much was lost. So that's how the students measured it for this particular plot. Yeah. But again, it depends on what resources you have. So if you can buy these commercial kits, that's a possibility. If you might be limited though, and if you're limited, you can ask about other aspects of the chemistry. For example, you can record pH. You can also look at things like turbulence. So how cloudy is the water. That can be done by taking a sample of the water, putting a card with different thicknesses of lines behind it and holding it up to the light and seeing which line is visible through the card. So there's some simple things you can do or some complicated things you can do depending on what resources are available. And the same goes for looking at organisms. So if you have a microscope, you can do what these students did and look at microorganisms. If you don't have a microscope though, you can dig into, and a lot of that, we actually had many students do this. They dug into the mud. They took the mud to the lab and they sorted through it to find different kinds of snails and different kinds of larvae of different insects. And we had pictures of the different kinds of insect larvae that lived in our area that they could compare them to.
So I think probably in all those scenarios, the best thing is to challenge the students with the question, what do you expect? What's your hypothesis?
And why do you expect it?
Que en el fondo el mensaje es siempre desafiar a estudiantes a que piensen y decirles, bueno, algo tan simple como tomar barro, ¿qué esperas tú encontrar en ese barro? Y un poco en el fondo desde ahí partir hacia la generación de una hipótesis para luego empezar a mirar qué es lo que hay en la muestra y si se comprueba o no ese postulado.
Yeah. That's the learning really happens when the student has to think, why do I expect this? And they start out, you have to coach them a bit because, the student will say, you'll say, well, here are the chemicals we're going to measure. Here are the kinds of biodiversity what we're going to measure. What do you expect? And they'll say, well, I expect less biodiversity with more pollution. That's a reasonable hypothesis, but what does that mean? Which of the chemicals that we're measuring is the chemical you think is most associated with pollution? what kind of biodiversity do you think will react to that increase in chemical? Are any of these pollution at all? So, an important thing to do as the students are working through this is to challenge them a little bit. What do you mean when you say this? I think we didn't measure algae in particular, but all of our students had hypotheses about algae. So they said, well, with more pollution, I expect to see more algae or less algae. I can't remember which you could justify both. And I said, well, we're not measuring algae. Of the things we're measuring, what do you expect? And they actually had to think a little bit more with that limitation. So, a lot of the, and this is what scientists do. Scientists say, well, what resources do we have? What is the interesting question? How can we use the resources we have to answer the interesting question? And it's important to remind the students that what they're doing is the same thing that professionals do. I can stop now actually and ask if, I can tell you about the next project in this class, but I can stop and see if you have any questions right now.
No sé si hasta ahora tienen preguntas de lo que ha comentado Primrose, o alguna acotación o algo, alguien quiera compartir algún tipo de experiencia. ¿No?
Okay, that's fine. I'll stop again in a little bit and check again. So we had, once the weather got very cold, the students finished, I should point out that they presented this plot. So the student group that collected these data presented this plot to the class, explained what they did, explained their hypothesis, their justification, and whether or not it was supported. We then had them do a project with this little organism. So this is a local zooplankton. It actually lives in the pond. Although we could have cultured these from the pond, but we decided to order them from a culture catalog. This is called a water flea. And the Latin name is Daphnia. It's very cute. It's so small that if you look at it under a microscope, you can see its insides. So you can see this little water flea's digestive system, and you can see its heart. And if I had a video, you would see its heartbeat. So you can actually observe a lot of its physiology. So we introduced them to it. We said, just look at it under the microscope, make sure you can see its heartbeat, make sure you know what that looks like. If the females have eggs inside of them, you can count the eggs. Now I'd like you to ask a question about how you might change its physiology. And we have many things you can choose from, but you're going to design an experiment. So in the pond, we had them do an observational study. In this, we're having them design an experiment. And we said, well, we have some chemicals, we have salt, we have drugs, we have nutritional supplements. Many of our students were interested in melatonin, which is a hormone that helps humans regulate sleep. We got a lot of these just simply from the pharmacist. And we also said you can change the temperature, you can think of ways to stress the animals, but we want you to form a hypothesis, and we want you to ask, how can I change the physiology? How would I expect the physiology of this animal to change? And at this point in the lecture, they're learning about homeostasis, and they're learning about osmoregulation. So they're learning about these processes by which cells maintain themselves, and they're thinking about how cells might be stressed. So they're applying what they learned in the lecture to the process in the lab. And so here they have a little bit more autonomy, right? We say, well, there's a lot of possibilities, but you choose. But you have to explain to me why. Why did you choose what you chose? And this is the really challenging thing. Again, the students have to say why. And once they've convinced the instructor that their idea is a good idea, the instructor gives them the materials they need. And they can do things like count, in this case, count the relationship between the salt concentration and heart rate in their Daphnia. So this again is a student plot. The student group presented this plot to the whole class at the end of the semester. And I like this plot because it shows that, in fact, they didn't see much of a relationship between salt and heart rate. So their hypothesis was incorrect, or at least it was incorrect for these Daphnia for the amount of time they gave them. And so they can talk about what happens when your hypothesis isn't supported, because that's also part of science. And in fact, one challenge is students feel like a failure with their hypothesis is not supported by their data. They say, "Oh, I got it wrong. I must not have had the right idea." And an important thing for the instructor to do is to make sure the students understand, no, you learned something. This is science. You learned something. You did a good job. This is how science works. We ask questions because we don't yet know the answers. So if we learned something, then we succeeded, even if our hypothesis wasn't supported. I find that this is a very challenging thing for students, that they really, their whole life, they've passed the exam, and they've been successful if they could pass the exam. And this is not an exam. This is a process.
Sorry, Primrose, there is a question from the audience, and so if you need that the students justify an hypothesis, have you taught the students to justify an hypothesis? Si se les ha enseñado a los estudiantes a justificar una hipótesis.
Yeah, so the question is, if I had talked to the students about how to justify a hypothesis, what a hypothesis is, and we start the semester by doing this. So we start the semester by practicing forming hypotheses, talking about what makes a good hypothesis and what makes a bad hypothesis. So a good hypothesis is testable, it's realistic, it's falsifiable. So we talk about that, and when the student comes to us with their hypothesis, we say, why do you expect to see this?
Sí, o sea, responde Primrose entonces que claro, lo primero que hacen es enseñarles a formular una hipótesis. Por lo tanto, luego de que evaluar de que esa hipótesis sea realmente testeable y preguntarles entonces por qué crees en esa hipótesis y cómo crees que puedes testearla o evaluarla.
Francisco, is your microphone on?
Yeah.
Okay.
Yeah.
All right. Just making sure. I think that's a good question. And I think that this actually, needs a lot of that kind of support. This whole strategy, if you just release the students and say, go do some science, they're beginners. That's why they're students. So you do need to teach them what that looks like, but then they get the opportunity to do it. So our entire first year biology introduction lab is really those two projects, and it's a whole semester, and those two projects take all that time. Sometimes they need to try things more than once. They present at the end of each project, they present their data. And so I wanted to stop now and ask if there were more questions from the group about how this works and how this feels and what the benefits or drawbacks might be of the strategy.
Eso, si tienen algunas preguntas respecto de esta experiencia hasta ahora.
Hi. I don't know if he...
Sí, parece que hay una... Rodrigo, ¿tú levantaste la mano o no? Sí.
Yeah. Hi, Primrose.
Hello.
Do you have audio? No.
Yeah, a little bit. I don't know if... well, I would like to know if the students, how you say it, compete with an idea, a project with your lab, or you can accept all the ideas and you work with all the students in this kind of science experiments?
Yeah, this is a classroom. So the context is classroom.
Okay.
This are lab classes. There are about 20 students in the lab. There are several sections. So all instructions, the full class is 150 students. So we divide them into 20 student sections. And each section is doing the same thing. The students, we don't accept every hypothesis. And that's one, yeah, we have to, the instructor challenges the student. Because, if the hypothesis is not falsifiable, for example, then there's no point in testing it. Sometimes they aren't practical. So sometimes the student says, we're going to grow the water fleas for 2 months and we're going to see what happens. And you have to say, well, that's great, but we don't have 2 months. You know, you have to do something that takes a week. You know, so there's conceptual problems and there's logistical problems, and the instructor and the students negotiate. Does that make sense? I think that's a very good question. And a part of me says, well, it'd be wonderful to just let them do anything at all. And another part of me says, actually, you need to push back for them to learn, because otherwise they can be a bit lost. So you need to say, you know, I won't support that hypothesis. I won't support you testing that hypothesis. And here's why. And here's, if you were to change it like this, I would support it. So go back and design a new one and come back and tell me what it is. I think there's one in the chat.
So Nicolas. Sorry, okay. Nicolas is asking whether you work in small groups or in general, like in mega groups, considering how good is this idea exchange between peers?
That's a really good question. We ask them to form groups of about 4 students. So in a class of 20, that's 5 groups. We have some flexibility. The students, of course, want to be with their friends. So we allow 3 or 5 students in a group if they like. 4 works very well. 3 works pretty well. 5, we find challenging. If you have a group of 5, 1 or 2 students often will sit in the back and not do anything. I should mention that one challenge of this strategy is that group work is a challenge and learning to work together is a challenge, and some students don't like it. Or you have the thing where somebody works very hard and other students don't work at all, or everybody wants to work but one student just takes over and does everything because they're just so excited. And this is something that the instructor has to help manage. We have some strategies for this. One thing we do is we ask the students to make an agreement before they start working together, and that they turn in the agreement to you. They all sign the agreement and they turn it into you. And the agreement includes questions like, how do you plan to handle it if somebody doesn't do a lot of work? How do you plan to handle it if somebody does too much work? What will you do if you have to meet outside of class to work on the assignment? How do you plan to do that? And they sign it so that when they will come to you and complain, so-and-so, they didn't do their job, you can pull out the agreement and say, well, in your agreement, you said you would talk to the person first. Did you do that? Okay, go do that, come back to me, tell me what happened. You know, and that can be a way to guide them towards working together. Let's be honest, working together is challenging. And they're beginners and they're not good at it yet. In another class, and we're still developing this. We've been doing this for 2 years now. In another class, an instructor tried having students role play good and bad group interactions. So, you know, okay, the student is not contributing. Why don't you show me how you would not approach them? And the students love it. They love acting. So they say, you know, how dare you not do your work? And okay, great. Now would you please demonstrate to me how you would do it in a helpful way? And they have to think about it. And so they practice that. So that's another strategy that we've been using. We also have them evaluate their group members, and we do it after both projects. So if after the first project, one group, they evaluate each other really poorly, the instructor can actually intervene and say, maybe this group isn't going to work for the next one. Why don't we move people around a bit? And that can help too. But I think group work is a really important aspect of actually all of the experiential learning we do. And it's a challenge. I think there might be more questions.
Yeah, I have a question.
Mm-hmm.
So you were saying that you have to accept the hypothesis, whether the group does it well or not. Do you have like a time set to Like, do you have like a week or two to make the hypothesis and then see if you accept it or not? Like, how do
Yeah.
you structure this whole lab process in terms of time?
That's a great question. So our labs are 3 hours long. So one thing we do is we will actually devote an entire 3-hour session to this. Or for the second project, we say, spend an hour looking at the water fleas under the microscope. Just look at it, draw a picture of it, see if you can't count the heartbeats. Now, and I'm sorry, there's a little bit of construction in the room next door. Now come up with your hypothesis and bring it to me. In practice, how this looks is that the students, they want to leave class early. So they come to you and they say, we have our hypothesis, can we leave? And you say, well, wait, no, tell me what your hypothesis is. Okay, no, you can't leave. You need a better hypothesis. And then, so that's also, a way, it depends, this would work differently if you have an hour several times a week, or if you have an hour every day. That's how we use that 3-hour session. I don't know, is that a satisfactory answer?
Yeah, so you're saying that you have like a whole class, like 3 hours for each phase of the experiment, like the
Yeah.
final experiment.
Yep.
Okay,
And
I see.
our semester is 14 weeks long. So it's about 7 weeks for the pond and about 7 weeks for the water fleas.
Okay.
And so we have the luxury where we can say, look, our hypothesis is really important. You're going to spend 1/7 of the time just on hypotheses. And then, the next week is measuring, the following week is measuring, the third week is measuring again because something went wrong in weeks 2, that we want them to take this time and we want them to take it seriously.
So how they measure things, they are the ones that are designing like the methodology or
Yes.
everything. Okay, I see.
Yes.
So
Yeah.
the teachers or the instructors don't say anything. You just let the students do what they think they need to do.
We do say some things. So for the water flea is a good example. When we first introduce the water flea, we say, here's a reading on how water fleas live. Here's a reading on their life cycle. Read this. Now look at the water flea. I'm going to tell you now that a lot of students choose to measure heart rate because it's easy to count the heartbeats. Many other students choose to measure distance they swim because that's also pretty easy to measure. If you want to measure reproduction, it's more complicated. You need to have a plan for what reproduction means. Are you going to count eggs, or are you going to wait until the eggs hatch? Maybe you need to come in outside of class to see how many hatched. Let's make a plan for that. So we give suggestions and we give options, but at the end we say, and you can think of your own idea as well. But if it's an idea that I didn't suggest, come and tell me what it is, and I'll tell you if I think it's practical.
I see. Yeah, thank you.
Sure.
So we have another question, and Cloud is asking, how much does the project last? And is there a certain time for each project?
Mm-hmm.
Yeah.
So because of the length of the semester, we devote about half of the semester to each project. 7 weeks, it's a little bit less than 7 weeks because we have a little bit of introduction at the beginning and a little bit of follow-up at the very end. So maybe it's about 6 weeks for each project, but we divide the semester in half.
Right. So it's only 2. There is another question, and Ruth is asking whether, so there is a difference between work in a group and to work in a collaborative way, right?
Yes.
Those are 2 things. It doesn't mean that, right? So the question is, when do you have this collaborative work? Like, how do you promote collaborative work?
Yeah. Gosh, that's a big challenge. And one of the things we do is we have the strategies I described earlier, where we ask students, what does collaboration, essentially, what do you want collaboration to look like? And we check in with them about how well they're collaborating. We ask them to write down how collaborative the work was. So they reflect on how collaborative it was. And we talk to them about examples of working together that's very useful and working together that isn't. One thing I found challenging is that often in situations like this, the students will sit down and say, okay, you do that, you do that, you do that. And we don't talk to each other and we bring it together at the end. I try to discourage that. And I tell students that the most successful projects are the ones where everybody talks together for every step. But it's an ongoing challenge.
There is another question, and is, so one of the problems that we have in the country and in Latin America in general is that public school, you can have 40 to 45 students in the room, right? So it's a large group of students, you know, going around and with a lot of energy. And so Karina is asking, like, how do you see this? How can you promote scientific thinking, you know, when you have so many students? And especially when you don't have resources. So let's say you have a room for 45 people, you don't have that much, you don't have budget, right? You don't have money to do it. So how do you see it on your experience? How can you promote it without money and just having that large group of students?
Yeah. I think, you know, a thing to do would be to think about, I guess there's two questions. There's the large group of students question and the money question. And I think they're a little bit, I think there's different answers to both of those questions. We found that as the number of students increases, that we've turned more to group work. That because the instructor can only spend, you know, only has a certain amount of time in the classroom, that we really rely on the students supporting each other as our classes get larger. You know, we see this more in our lectures. We have a legal restriction on the number of students in the lab classroom, and that is 24. So that's a fire department restriction, essentially. In the lectures, though, they can be very large. They get to be 70. And so we rely a lot on the students supporting each other, and we try to give them a framework for that. You know, how does that group work and collaboration work? And I think that's, you know, that's a good way to deal with having a lot of students. How to deal with not having so many resources. I think, you know, the question to ask perhaps is, what do you have? A lot of field research can be done even in a city. For example, maybe yours, you know, the city you live in, that a water, you know, a big pond like the one we worked in isn't available, but you can ask about, you know, biodiversity in soil, in a garden. You can have students ask questions about birds that they see in their local area, and ask about, you know, conditions, climate conditions. A really nice resource that can be used as public data. So online, many countries and many communities will post historical weather data, for example, or data about air quality or data about water quality. And you can relate that to the students' questions. So you can say, well, in our city, maybe students live in different neighborhoods. So you can have the students perhaps count the birds they see in their home neighborhood, and then look up records about, you know, the air pollution in that neighborhood, and say, well, what is your expectation? How do you justify it? I don't know if that was a satisfying answer to that question. Public data is really, it's really great to work with. Scientists work with it, and of course, we always want to connect with you are doing science. We're doing the same thing scientists do. I use publicly available weather data for my own work as a scientist, and so having students use those kind of data is doing science.
Carlos, puedes prender tu micrófono, por favor.
I'm sorry to be in the bed because I am in a sick leave.
Oh no, I'm so sorry.
It's not because your presentation is boring.
I hope you feel better.
Yeah, a little bit better. So my question is from the point of view of the teachers. So, you know, probably in our university, many of the people who have to taught in any course were not teachers, were scientists in the circumstance to have to teach. That's it. Yeah, this is the whole preparation. But I wonder, even in your setting, what are the more usual questions or help the teachers ask to each other to improve the teaching in your college?
Yes, so in our college, in fact, we are in the same situation. Even though we are mostly teaching, the balance between teaching and research is weighted towards teaching. We are not trained teachers either. We are trained scientists. And that's an advantage and a disadvantage. The advantage is that it means that we're thinking about ways to bring science into the classroom a lot, and it's inspired by our own research interests. The disadvantage is, of course, that we are not trained instructors, and that is a skill that we sort of have to pick up on our own. And as a department, the way we're dealing with this, and I'm so sorry, I worry that you might hear the construction noise outside. But the way as a department we deal with this is that we've actually been seeking out kind of continuing education for our department. So we have been collaborating with people in the education department. We have been collaborating with outside consultants. And our institution is specifically focused on diversity, equity, inclusion, and belonging in science. So that's an institutional priority. And so that's and part of it is that we got this grant to support this, that we can afford to hire outside consultants. So we also have we had a reading group where we would read education literature to find best practices in the literature, and then have a essentially journal club on those. So that's how we have been working on it. It's a big time commitment, and if you work at a research institution, there's a trade-off because it takes time away from your research. We're lucky in that our priority is teaching, and therefore it's time spent improving teaching for us is time that's rewarded by our institution. I don't know if that's the kind of answer you were looking for.
Yeah, thank you.
Thanks.
We are in the same page, in the same trouble.
I'm sorry, another question? Oh, so I'll continue. And I want to talk about this new series of courses that we've been offering for second-year students called Research Experience in Science Communication. And this is a full class where the student asks a question of their choosing on the topic of the class. And we offer several topics every semester, so the students can choose the topic from among the topics that are offered. This is student-driven. There are several choices of topics. It's required for second-year students. So if you want a degree in biology from Wheaton College, you must take this course in your second year. And you also must take a course called Science Communication. In Science Communication, the students learn to write a scientific paper and present a scientific poster, just like working scientists. And so we have a poster session at the end of the semester, and we make it a big celebration. We have coffee and cake, and we make the first-year students go to see the posters, and the first-year students have to ask questions at the posters, or they get a grade for that. And we want the second-year students to really celebrate the work they've done. And I'm going to tell you about my experience doing this. I ran this course where the topic was toxins that microbes secrete, specifically single-celled fungi, to kill other microbes or other single-celled fungi. And I spent the first week of class introducing students to the topic. And so I'll just introduce you very quickly. I'm showing you a Petri dish with some colonies, and I'm showing you some very larger colonies that are spotted on top. Some of the larger colonies produce a toxin that kills the smaller colonies. And so you can see that right here. And I presented this to the students, and I said, you have an entire semester, 15 weeks, to ask a question, design an experiment, conduct an experiment, and then present a poster and write a paper on this topic. And so one example of a very successful student group, this group had 3 students in it, and you can see them in the photograph, is they said, does the yeast that produces the toxin have difficulty growing in a stressful environment? And I said, okay, what's your expectation? Their expectation was that toxin production would make it harder to grow if you were stressed. And I said, great. What does stress mean? And they thought they had to think about it. What does stress mean? And in this process, we're having them read scientific papers so that they know what the literature is that they're building on. We want them to ask a question that hasn't been answered before. So this is a little bit more sophisticated than the Pond and the Daphnia projects we had them do, in that they have to use the literature now to design the project. There's less involvement from me. I still get to give them feedback, and I still get to tell them, I'm sorry, your idea is a great idea, but it's not practical. We only have 15 weeks. I said that a lot in this class. That they get mentorship, but they get less guidance from me. And it helps them to take more responsibility. So I just kind of want to take you through what this group did. They grew yeast cells and they measured how much they grew. The stress they decided on was low nutrients, so a small amount of food. At the end, they produced this plot. And while I told them how to produce a plot, the students produced this plot. And in this plot, they had 2 treatments, high glucose and low glucose. And they had yeast that produced the toxin. The yeast that produced the toxin are called killer. They found that when the yeast were stressed, the killer yeast did actually a little bit better. And when they weren't stressed, the killer yeast did a bit worse. So they didn't support their hypothesis, but instead they got a different and more interesting story. And with that story, they made this poster. And they presented this poster to the entire department and to all their friends. So we invited their friends. Again, there was coffee and cake so that people would come and drink coffee and eat cake and look at this poster and ask the students questions about it. And the real lesson for the students during this process is that science is challenging. This particular group, they were growing microbial cultures, and every week they came back and their culture had some contamination. And I said, I'm so sorry, you have to try again. And they did until they got it right. So they had to do it again and again and again. Sometimes a group member would make a mistake in the measurements, and we'd talk about how to handle that gracefully, because it's easy to get angry, but in fact, human beings make mistakes. So how do we handle that? This necessitated repeating work many times, and problem solving. Why did you get contamination? What went wrong? What was the mistake, and how do we avoid it next time? Students also often had the problem where if their hypothesis was not supported, they would think that they had done something wrong. And so an important lesson is you asked a question about nature and you got an answer, so you learned something. Even if your first guess was wrong, the important thing was that scientists learned something about nature. And this is an emotional process, because now it's their project. It's not, I give you some water, please have fun. It's, no, you have gone into the literature, the scientific literature, you told me what was missing. You'd asked a question, posed a hypothesis, this is yours. And so it becomes an emotional process. And the 2 most common emotions that we experienced were frustration and excitement. And so it's a way for students to learn how to deal with emotions when they're doing scientific work. And I'd say maybe frustration and excitement were about equal, about 50-50. And it's good, you want that. You want this to be a rewarding process. My department is collecting data on the student experience in these classes with the intention of eventually publishing them. I don't have access to the full data set, and I don't really want to step on my colleague's toes who's writing this paper, but I do have some of the data on the time I taught the course, and I figured I'd just tell you about some data points. So we surveyed students before and after the course, and we included the statement, I feel like I belong in a STEM discipline. And the students increased in their amount of agreement with that statement. They also increased in their intention to go to graduate school. So these are bachelor students who are trying to decide what they want to do when they grow up. And part of this was saying, this is what graduate students do. Graduate students work independently. Sometimes they collaborate in groups. They're asking their own questions and they're taking responsibility for their own decisions. And that this felt more attractive to at least the cohort of students I worked with. And that they also stated that they formed new connections, that they made friends and they learned how to work together. And so those were positive impacts of this class. Again, we're still working on making it better. We're still growing and developing this class. Having spoken about that, I'll stop again and ask if there are any questions.
Preguntas que tengan hasta ahora de esta siguiente parte. Parece que por ahora no. Si no, las van escribiendo y podemos ir comentándoselas a Primrose.
Okay. In that case, I'll be very quick then. The last thing I wanted to talk about was our microbiology lab. This is an introductory lab. They've taken introductory biology and they're learning microbiology. They're mostly first and second years. So I want to talk about this, this type of course. This is called a course-based undergraduate research experience. The courses I just presented are examples of this, but there are some that are published. And let me quickly just copy a link into the chat. Let's see here. This is the chat. This is a book on this kind of experience. These are courses where the students do real research. And unlike in the course I showed you earlier, where the students communicate their work with the local community, in course-based undergraduate research experiences, students communicate their work to the broader community, to the whole world. And so we tried out one of these in our microbiology class. And this was designed by researchers at a different school. And the students were to collect soil and measure antibiotic resistance in the soil. And I won't go into details of how that worked because we don't have so much time. What I wanted to show you is how the students are taking their data and they're contributing the data to a database that anybody can access online. And I'll just copy and paste the link to that right now. Let's see here. There's a lot of projects like this that exist, and in the link here, there's actually a whole list of them in that textbook, where you can have your students collect data on a question that's not your research question, but that's the community's research question. And the students can share their data with the broader community, usually on in some kind of online system. I prepared these microbiology students for this work very carefully. I told them, you're going to do work that is actual science that's going to the community. So you need to learn these skills first. And this is a way I got these students working on these these important skills for any microbiologist. They had to learn lab safety, they had to learn technical skills. There's a lot of math in this problem, so they had to learn the math. And then again, they posed their own hypothesis about the data. In this case, it was, where locally do I expect to find a lot of antibiotic resistance in the soil? And we talked about why you might expect to find antibiotic resistance in the soil. So they still have ownership and responsibility, even though they didn't design the experiment. The experiment was designed by these outside colleagues. Their data goes to the outside colleagues, and it goes to the broader global community. Now, I noticed for this particular activity, there are no data points for South America. And so I would love it if some of you would like to join this, because I think it's a great project. And I think that the organizers would love to reduce some of this North American bias that you see on this map. So I just wanted to kind of tell you about how we're incorporating experiential learning in our classes. Our goal is to have experiential learning in all of our laboratory courses, because we think this is so important for students, especially because of the sense of belongingness that it gives students. It gives them the responsibility, it gives them the initiative, and it helps them to think, oh, my ideas are good ideas, they're worth testing. I had, we don't have time, but I had an activity where I said, it would be interesting to ask you how you could imagine using these kind of strategies in your own teaching. So I might encourage you to do that after this session and share your ideas with others in this class, to think about how you might see that working for you. Let's see here. I think, I could talk more, but I think I'll stop and ask for more questions. Those are our 3 experiential learning classes that I really wanted to highlight. If there are any questions.
No sé si hay más preguntas para Primrose de lo que ha mostrado hasta ahora, o algunos comentarios, algunas cosas. Felipe.
Yes. On the research experience and science communication courses, is there any, because you made it on toxins, right?
Yes.
So is there any consideration on students' interests, or is it like just toxins, like the teacher says, okay, we're gonna do it about toxins, and you see what you do?
Yeah, that's a great question. So in our school, we offer 2 or 3 sections of this per semester. It means that a second-year student has 5 opportunities to take this class, twice in one semester and 3 in the other. And we publicize the topic. So the idea is that the student picks the topic that they find the most interesting. It doesn't always work that way, because the student really picks the topic that fits with the rest of their schedule. They say, well, I have sports and I have some other class, so I'll pick this one because it's on Fridays at 4 o'clock or whatever. But the idea is that we give them the opportunity to choose. And then once they've chosen, we say, okay, well, you chose the one on toxins. So here's the toxin story. Now here's some papers you can read, or here's how you find your own papers. Now develop your hypothesis based on that. You can make it broader, but it becomes impractical if it's too broad. I'd like it to be very broad. I'd like the students to come in and you say, welcome scientists, do some science, but it ends up being impractical.
Yeah, thank you.
Sure.
No sé si hay más preguntas para Primrose. Parece que no. Rodrigo tiene otra pregunta.
Again.
Sorry about the noise.
No, don't worry. I hear very well. Thank you for the link. It's really interesting, and I'm going to try to see, maybe investigate something about that. I have a little question. In your opinion, with your 3 steps in this, your experience, science experience, is maybe one of them more challenging? Maybe the experiment, or the communication part? I don't know if you understand.
No, the problem is there's some outside noise, so I didn't hear you, but they just stopped. So tell me again very quickly.
You think that in one of your 3 steps, one of them are more challenging, maybe the experimental part, the communication of science, or?
I see. Yeah, they have different challenges. A big challenge is the group work and the collaboration, and this was highlighted by previous questions. The science communication is challenging, but of course that's something that it's part of our job to teach students, right? How do you write? How do you communicate? Yeah, I don't know. I don't know which is the most challenging, but I think every section has some challenges. The hypotheses are challenging, often because students just don't know what a hypothesis is, and they don't one thing they have to learn is that you build hypotheses based on the information you already have. So you have to go and look for that information. You know, I say, this is what a toxin is. And then I say, here's how you find scientific papers in the literature. I'd like you to find one yourself, read it, tell me what it says, and tell me what the next question is. That's something they don't know how to do. You have to teach them how to do it. And that can be challenging because they think they know. They think they know all of this. And you know, that once they try it, they realize they don't. One thing that I've found very successful is that I make them plan every step. And I say, imagine you got drunk last night and you're hungover and you can't think and you can barely stay awake. You need those instructions for yourself that you already wrote so you don't lose track. And I give them feedback based on that. Like, could I imagine doing this if I were drunk, you know, or something like that? They think it's funny if I talk like that, but also it's important because when we make plans, you know, we don't anticipate everything. And so we have to be really, really on top of things and be as prepared as possible. That's something that takes practice. They don't get it right the first time. And the nice thing about this is it's an opportunity for them to practice. You know, the stakes are low. Nobody's going to fail the class because they made a single mistake. So they have the opportunity to make mistakes. And what we're looking for is, well, you know, at the end, what do you have to say about the mistakes? Or what do you have to say about what happened? You know, that's the thing they're graded on. And that can be really, really nice for them.
So some people's got comments like Nicolas is saying that he congratulates you about sharing this and doing this effort, trying to drag the students into science, right? And also that's the challenge. He says that's our challenge as teachers, you know, to get them interested in science. And some of these experiences and strategy are really valuable for us. So they're really happy.
Well, thank you so much.
Ok, bien, bueno, dejamos hasta acá la sesión de Primrose. Muchas gracias a todas las personas que asistieron a esta charla. Ojalá que haya sido para algunas personas un buen ejemplo de cómo seguir adelante y de cómo atraer a diferentes estudiantes hacia la ciencia. No olvidemos nunca de que se trata de hacernos preguntas y poder responderlas, y eso es algo que ustedes como profesores, como profesoras pueden hacer desde el colegio y hacia el futuro. La verdad es que está todo en sus manos y ojalá lo puedan lograr. Thank you very much, Primrose, for your time, for your experience. People is very happy about this. So thank you very much.
And thank you so much for your attention and for coming. There are challenges when doing this virtually, so I really appreciate your interest, and thank you so much.
Más adelante voy a subir este video a YouTube para que les quede para más adelante. En una de esas pueden activar también la traducción al español y así les pueda servir algunas cosas que quizás quedaron por ahí que les interesaron y que les gustaría repasar. Mucha suerte en el trabajo que les queda a futuro y cualquier cosa, ustedes tienen mi correo, tienen también podrían tener el de Primrose, lo que sea que necesiten ahora en el futuro, siempre estamos disponibles. Así que muchas gracias Camila por organizar esto y por ayudarnos con toda la logística y a todas las personas que hoy día asistieron. Muchas gracias. Chao, chao.
Very nice of them.
Thank you very much. Nice experience. Thank you very much.
If you're hanging back because you have a question, I'm happy to answer it.
Muchas gracias, Camila. Salió todo bien.
Sí, vi que salió todo bien. Ahí estuve tratando de ayudar con algunos micrófonos que sabían y
Sí.
la traducción. Y bueno, el video cuando me lo pueda mandar lo subimos al YouTube de la facultad, entonces ahí lo puede compartir. Thank you.